The cells in our bodies store their genome in a single compartment called the cell nucleus. The genome itself, however, is distributed across numerous individual and independent storage units which are called the chromosomes. Now, when our cells divide into two, then they undergo a process called open mitosis, and this means that they fully disassemble their nucleus in order to allow proper chromosome segregation to the two daughter cells. This also means that the cell actually has to reassemble the nucleus at the end of every cell division, and this is a rather difficult task because it's very important for the cell that all chromosomes are targeted to a single nucleus at this step. It's difficult because the cell actually cannot count the chromosomes and they all look the same. So we asked which mechanism actually is involved in ensuring that the chromosomes are unwrapped as an ensemble, and what prevents chromosomes from being unwrapped individually by nuclear envelope membranes. In order to find the key factors that are involved in this process, I performed a high content screen and I identified BUF, a small DNA binding protein, as a key factor in this process. Now BUF binds to the surface of the anaphase chromosome ensemble just before they're enwrapped in nuclear envelope membrane. And interestingly, what we observed is that BUF forms numerous cross bridges between neighboring DNA strands and ultimately between neighboring chromosomes, which stabilizes the entire anaphase chromosome ensemble. When we reconstituted parts of this process in a test tube, then we observed another very interesting property of this BUF DNA meshwork at the chromatin surface, and that is that it actually provides a barrier against macromolecular structures. So this explains how DNA cross-bridging prevents chromosomes from being enwrapped individually, and how a single nucleus is actually formed from a set of mitotic chromosomes at the end of cell division. So this provides an elegantly simple solution to a fundamental problem of open mitosis.